So what you're looking at is actually a three-dimensional reconstruction of mitochondria in the brain tissue of, in this particular case, it's Alzheimer's disease mice. So why mitochondria is on the first slide, I hope you will understand at the end of my talk. Oops. So the focus of the lab is actually on identification of early molecular mechanisms of Alzheimer's disease, development of biomarkers, early methods of diagnosis, and therapeutic interventions that could restore damage acquired to brain tissue and neurons during the development of Alzheimer's disease. It is now well accepted that the beginning of Alzheimer's disease occurs many, many years, up to 25 years before the onset of any cognitive symptoms. So there is absolutely no treatment that modifies the disease, and there is no methods of diagnosis. Major reason is that 99% of Alzheimer's disease is sporadic, and we really do not know what happens in the organism, in neurons, that leads to this devastating disorder. I do not know whether anybody watched yesterday the CNN piece on Alzheimer's disease yesterday. It was awesome. So I'm not really going to spend much time talking about pathology of Alzheimer's disease, but the work I'm going to present you today is actually focused on really development, the early methods of diagnosis in plasma, because I think it's very important that the source of the metabolites will be readily available for multiple screening and testing. So Alzheimer's disease is characterized by extracellular amyloid plaques, intracellular phospho-tau, and you can see the devastating impact of the disease on the brain tissue. Mounting evidence suggests that mitochondria and abnormal brain metabolism is involved in Alzheimer's disease. One of the most compelling evidence comes from the FDG PET that suggests that in people with mild cognitive impairment, the glucose utilization is severely impaired. So that was our first clue to kind of start looking into what's going on with metabolism in Alzheimer's disease. And the very first study I'm going to show you kind of got me into the metabolomics field, so I cannot, even if it's published, I cannot omit it. So we have multiple animal models in the lab, and this first study was designed to ask the question, can we see, using metabolomics profile, any early changes in the brain tissue of these transgenic animal models that have familial Alzheimer's disease genes? So I said that 99% of Alzheimer's disease is sporadic, but we're lucky to have 3 genes that if mutation occurs, these genes, the mutations lead to the development of Alzheimer's disease. So we have all these animals in the lab. So we'll look not at one particular animal, but 3 different models. And when we study expression of metabolism, metabolic signatures in the brain tissue of these animals in pre-symptomatic stage, so very early, before these animals have any cognitive impairment, what we found that these animals had a very distinctive metabolic signatures, but all of these changes were associated with energetic stress. What was also interesting, and that actually I hope laid to the grant that we're working on right now, we found that in female mice, the metabolic alterations were more pronounced than in male mice. So when we look at specific panels of metabolites in all of these different animal models, we found that metabolites that relate to specific mitochondrial functions, including TCA cycle, were affected the most. So the conclusion of this study was that metabolomics could help to identify signatures of energetic stress associated with Alzheimer's disease in pre-symptomatic stages. So the extent of metabolic changes was more pronounced in females. Metabolic changes were consistent with actually changes detected in humans, and metabolites that were used to look at changes in patients suggesting translational potential. And these changes were all leading to abnormal mitochondrial function. So the second study was designed to confirm our observations in humans. So working at Mayo Clinic, I have an access to Alzheimer's Disease Research Center collection of human CSF and plasma, and we conducted pilot study where CSF and plasma from the same individuals who are cognitively normal or have mild cognitive impairment, which is preclinical stage of Alzheimer's disease, or Alzheimer's disease, with 70% of males in this group were subjected to non-targeted metabolomic profiling. What we found is that we could separate the groups pretty well, and we could separate the groups in plasma and CSF. And to my knowledge, the results were published in 2013. To my knowledge, that was one of the first studies that consistently compared the same individuals, which were very well characterized in terms of disease diagnosis and other parameters such as amyloid beta and so on and so forth, that we had a very nice separation in the plasma. So the next question was, what are the major metabolic alterations that could help to validate plasma as the reliable source of biomarkers? So we compared the pathways that were most affected in CSF and in plasma, first in MCI patients. And you can see in red the pathways that were affected to the same extent or the same pathways in plasma and CSF. So this is mild cognitive impairment, people compared to control. But if you will look at what pathways were affected the most, you can see that in CSF it was TCA cycle, and TCA cycle was almost on top of pyramid in plasma. Other mitochondria-related or energy-related pathways were affected as well. So what about AD patients? You can see now that there is much more pathways that are significantly affected in these people, and some of these pathways, again in red, the same pathways in CSF and plasma, and you can see that there are pathways that are involved in diabetes, but now lipids are on the top of the pyramid, but still we have TCA cycle and mitochondria-related, energy-related pathways affected as well. What we concluded from this study, so this is actually differentiating pathways when we looked at all three groups in CSF and plasma. So what we concluded from this study is that metabolomics offer novel approach to identify alterations in pathways in AD patients. It allows the identification of expected, non-expected individual metabolites, but most important that plasma really very well to actually up to 60% recapitulated changes detected in CSF, validated it as a good source of biomarkers. So the next study actually was applied to the same group of samples, but now we increased the number of samples, making each group 50-50 between males and females. This study is ongoing, so I do not have right now the results to show you what differences we can pick up in males versus females, but we're doing this right now. So we applied targeted approach to see whether we can now really nail down the particular metabolites based on our non-targeted approach. So again, with this targeted approach, when we looked at lipids, amino acids, TCA cycle, and fatty acids, we were able to separate the groups pretty well in CSF and plasma. And in biomarker panels, now you can look at the most important 30 of the most affected metabolites. With the asterisks, you can see TCA components of the TCA cycle. So in the MCI patients, there were ceramides actually in CSF and plasma on the first most important rank, but TCA cycle components were still there. In AD patients, you can see that again we have in CSF lipids on top, but also components of TCA cycle. But all of these TCA cycle components became the very first most affected metabolites in plasma of AD patients, which was very encouraging. And you can see a lot of abbreviations. These are actually clinical parameters that include cognitive tests, results of scanning, results of amyloid load. So this is a very interesting study that we keep analyzing this data to see how metabolic changes could correlate with other parameters that we have for these patients. So when we looked at most affected metabolites in plasma now from all three groups, you can see that components of TCA cycle came on top. So again, being at Mayo Clinic, it's very beneficial because we have a lot of talented people who work with clinical applications that could help to analyze the data and make analysis simple. So Dr. Rinaldo, who is working at Mayo, developed the clinical application called CLEAR that was developed originally to identify metabolic disorder in newborns. So that was supported by 2 consecutive NIH R1 grants, and right now it's a web-based application. When they run MS data on the blood of newborn, they can upload this data, the panels of metabolites, wherever they are. So this program now involves 46 countries worldwide who contribute their data. And the more data you put in, the smarter the program becomes. The major advantage of this program is that it develops the ratios and particular changes at metabolites that brings clinical importance to your data. So we apply this CLEAR program to this targeted metabolomics that we did in AD, MCI, and control patients. And the training set was designed, all parameters were uploaded, and you looking right now at this plot where you can see AD patients in purple, blue is MCI, and the red is a subject that the system placed in the exactly correct position based on the training set. So we're very hopeful that application of this program, when we will validate it, could offer metabolomics community huge advantage of analyzing the data, being where you are, and kind of opening very efficient and easy way to analyze the data. It could be analyzed from animals, from different tissue. It's not limited to CSF and plasma. It can actually open enormous opportunities to compare the results in different tissues, different animal models. So you can also enter any clinical parameters and compare the levels of A-beta versus changes in this or that metabolite. It's incredible. I mean, it's a lot of work, and as always, this is the limiting factor, but we're slow but for sure trying to apply this program to our data. And the final study that I'm going to really run through was how can we verify what is going on with this TCA cycle. So what we did, we actually applied stable tracer technology, flux, to examine flux through the TCA cycle using glucose and glutamine tracer. And what we found, and really wrapping it up, that there was no changes in the enrichment when we applied the glutamine, but when we applied glucose tracer, we saw, and you can see the statistics around the TCA cycle, that females with Alzheimer's disease were specifically affected compared to males. What was interesting, when the data was pulled together, males and females, we did not see any statistical differences. We only saw it when we analyzed males and females separately. So this is one of the very important conclusions that I'm going to say, that metabolomics data could provide great opportunities to study what's going on, to verify different aspects. What we're doing right now is we have multiple models or assays to study mitochondria function. So we're now figuring out what's happening in this fibroblast in terms of mitochondria dynamics and function. This is one of the examples that when we looked at the same fibroblast, where there was no changes in glucose, enrichment from glucose, compared to females, we saw that membrane potential was specifically decreased in female fibroblasts compared to no changes in males. So there's interesting general biology changes in males versus females that actually translate into metabolic alterations. So we're also going to use seahorse, which I'm not going to talk about because of the time. And we're very hopeful that now with these multiple assays, multiple applications, building our expertise, we can really move the field forward, trying to develop understanding of the early mechanisms that are involved in Alzheimer's disease, trying to develop cure, and developing plasma-based, blood-based biomarkers. So all of these people on this slide are important, and I think, again, the sponsors, without NIH funds, we will not be able to do that.
So this talk is open for questions.
The people from Biocrates several years ago presented data on how plasma metabolites change with aging. And one of the highest ones, I think it was number 4 on their ranked list of most changing with age, was alpha-ketoglutarate. So a question in what you've done is, did you extract out all the effects of aging that you might have encountered? Just looking at the mean ages, and they were a little bit different, I noticed on your last slide. Might be a factor that's in there that maybe is not a difference, but it's an aging effect.
This is an excellent question, because aging is one of the most significant parameters contributing to Alzheimer's disease. I was just sitting, listening to the talks, and that was exactly what I was thinking, that I have to go to our aging center, and got together with them to basically do that and see how much of the changes that we see in Alzheimer's disease patients can be recapitulated in just normal aging. But again, you have to do longitudinal studies along with cross-sectional. It seems that we're very successful. I was the skeptic one. When we started with 15 people, I was thinking we will never see group separation. Forget it. I mean, we have to have 150. So we really can do that with reasonable numbers to see changes. So yes, absolutely, you're right. It's a great point.
Thank you.